It's always a pleasure to come to this meeting. I feel like I'm coming home when I'm here. These are my disclosures. And what we're doing with the molecular microscope is basically receiving biopsies, processing them on microarrays, subjecting them to predefined machine learning derived algorithms, and then characterizing their phenotype in two big dimensions, the rejection dimension and the injury dimension. These are diffuse field changes in the biopsy, so we can do that. So the project provides us with a large population of indication and protocol biopsies characterized in the clinical, histologic, molecular, and outcomes dimensions. And that is really what I'm talking about today. I'd like also to point out that we've got a lot of experimental mouse transplants, and a lot of the background conclusions that we have characterizing the biology comes from those, including knockout mice. And this is the Edmonton team. And this is the set of collaborators. There are so many of them, it's hard to thank them all, but much of the biopsy material we have, it comes from European centers, and I'm very grateful for that. These are some of the clinical trials that are ongoing in kidney, heart, lung, and liver. Of course, all of these are from patients it's in IRB-approved protocols and from consented patients, and we always must remember that we owe a debt to the many individuals who've contributed a piece of themselves to this project. So let's talk about first of all, about the molecular assessments. Then we'll use the kidney as an example, although we have a lot of data now in hearts and lungs, which sort of corroborates these principles. So do we believe in molecular assessments, and why? Well, first of all, these are the top 10 reasons. When predicting a phenotype molecularly or conventionally, the molecularly usually wins if you've got a hard phenotype like survival. We've done that many different times. Microarrays use more features, far more features, 50,000 features, as opposed to the features used in a conventional assessment. The molecular features are continuous and not semi-quantitative or ordinal or simply dichotomous like histologic features. We're not just talking 0, 1, and 2, we've got numbers. The molecular features the molecular-based classifiers are trained on the clinical features, the histologic features, and they incorporate the molecular features, which actually makes them more powerful. Biological relevance of many of the genes has been established. The analysis has provided already evidence for updating BAMF, so basically the recognition of the importance of C4d negative antibody-mediated rejection came from the molecular analyses. So we use probabilistic assessments and not categories, and we obey the rules. When you have an algorithm in the computer, you don't get to say it depends. We use machine learning, and one of the things about machine learning, and I'll show you this, is that we can do simulations to see what the error rate would be and what the machine learning does with an error. So machine learning using histology assessments as the training class can overcome errors in the histology assessment and still get the right answer. Let me show you what I mean. These is from Jeff Reeve. Supposing we have antibody-mediated rejection that we're very happy about, and a similar number of cases that we're very happy doesn't have it. So now we have a 100% label correctness, and we do a classifier then to predict the performance. That classifier is essentially perfect in another set of biopsies in identifying antibody-mediated rejection versus none. Okay, so that's perfect when the labels on the training set are correct. If the labels are random on the training set, the classifier will not identify the phenotype, obviously. If the labels are 10% correct, 10% erroneous, and we've made them deliberately 10% erroneous, you still get perfect identification of the truth. 20% is still perfect. 30%, you're still getting an overall assessment of the truth. So in other words, machine learning can overcome errors in sample labeling. And it's superior for predicting survival. So in this analysis that we published recently, we did C-statistics for predicting using the histologic diagnosis, the archetype cluster assignments, so grouping the biopsies, or the actual archetype scores. And what came out was the actual molecular scores for the individual biopsy predict the survival of the individual patient. The molecular groups they could be assigned to are a little less good, and the histologic groups are worse still. So this is actually getting toward the point of getting a precision assessment of the individual patient on the basis of their data. If we have antibody-mediated rejection identified molecularly, and we look at the C4d positive or C4d negative, then the C4d negative is also, the DSA positive versus DSA negative, the DSA negative are also at risk. And the histology rule precludes them from actually having a diagnosis. So that those patients are being missed in the current algorithms used for histology. So this is a way that molecules can correct errors in the histology algorithms. Now, the rejection dimension, if we look at the rejection dimension, we start off, first of all, the way we do it currently is we start out by generating multiple classifiers using a positive class and a negative class for antibody-mediated rejection, T-cell-mediated rejection, and then also for lesions, T lesions, I lesions, PTC lesions, etc. So we generate those using cross-validation methods, and then the only thing you ever see from us is the cross-validation assignment in the test set. And so each biopsy eventually gets a test set result using these cross-validation methods. So we end up with characterizing each biopsy in terms of 7 individual molecular measurements, each of them trained through 12 different machine learning algorithms, so 84 equations. So that gives us a matrix of the biopsies, and the features of each biopsy then can be represented in principal component analysis, and that's what this is. As you can see, there are not distinct groups, and each biopsy has a position in data space. The antibody-mediated rejection up here, T-cell-mediated rejection here, an interesting tail of severe T-cell-mediated rejection here, and no rejection over there. So that's the rejection phenotype at the molecular level. Now, when we apply archetypes, archetypes are a way of extracting the actual precise molecular phenotype into categories which would be actionable, and that's what we're trying to do. So the archetype is actually identifying the typical cases, the idealized typical cases that minimally would account for the variance in the data. So when we apply the archetypes, what we do is this. We generate the archetype models. We then look in a scree plot at how many cases, how many different idealized cases there are to optimally explain the data. And we came up with 5 or 6 optimal phenotypes, which could be assigned as archetypes. And then each biopsy can then be given the archetype score for each of those features. In each biopsy, the scores add up to 1. So this individual biopsy has a score for mixed rejection of 0.89. That's what it is. It also has scores in the other dimensions, all adding up to 1. This biopsy has an individual score for full house antibody-mediated rejection, and the other scores all add up to 1. So that's the archetype of that biopsy. It describes that individual, and that's really precision medicine. That individual is being assigned those archetypes. So this is what we get from that analysis. This is how we move from the overall smear of the data for all the individuals into categories. No rejection, T-cell-mediated rejection. This interesting T-cell-mediated rejection, which is mixed and associated with non-adherence. Early stage, fully developed, and late stage antibody-mediated rejection. And those 6 archetypes seem to be sufficient to explain the variance across the data as categories. They're also distributed in time. In the overall population, this is 1,208 biopsies, we see a peak of early stage ABMR as one of the first presentations, almost always in people that were known to be at risk for having a pre-existing DSA. Then we start to see a late peak of early stage ABMR, peaking around 18 months, followed by fully developed ABMR, peaking around 4 to 5 years, and late stage ABMR peaks after 10 years. That seems to be something about the natural history of the ABMR problem in the population. It isn't exactly a time course for an individual patient, but it's telling us something about the evolution of the disease. T-cell-mediated rejection peaks in the second half of the first year and has a second peak in the non-adherence period in the second and third years. Now, when we apply the archetypes to the principal component analysis, we see the archetypes distribute as early stage, fully developed, late stage ABMR, TCMR, this TCMR mixed non-adherent phenotype, and no rejection. And we turn that and look down the barrel of principal component 1 so we can see principal component 3. We see The principal component 3 is being driven by early stage, fully developed, and late stage ABMR by the natural history of the ABMR disease. So now we're moving toward actionable categories from highly granular data, and the categories go to the rejection assignment, and we're still working through this. So we think we need a possible to explain the data. So possible TCMR and possible ABMR. Now we can examine that with a method called Kodama. Now I'm not going to try to explain it because I don't understand it. Jeff Reeve does these analyses. But what we're doing is, this is a method for saying, are there groups in the data and have you adequately explained the groups, going back to the original molecular phenotypes. And this says you do have groups. There's TCMR. There's the TCMR ABMR mixed non-adherent phenotype. There's no rejection. There's early, full house, and late ABMR separating in space. And they also separate in the second and third dimensions as well. But it also says there's a group called possible ABMR and a group called possible TCMR. We don't exactly know what they are, but they are different from no rejection and different from the fully developed phenotypes. So this is a getting toward how to establish categories which could be actionable. Now, when we look at discrepancy analysis, and we won't put you to sleep with the details of a discrepancy analysis across 1,200 biopsies, but what we see is many of the discrepancies occur in areas where the BAMF rules have been known to be weak and contentious. So without going into detail, we now see, yes, the discrepancies can be errors either in histology or molecular, but a lot of the errors are probably in histology. Okay, one of the interesting things that I've found in my career is looking at injury. And injury is a remarkable phenotype. All organ transplants are injured in the process, and they're profoundly injured in the process of the donation implantation period, and they never forget it. So we see the acute kidney injury response, which is the response which occurs within hours to days in the transplanted kidney. And we see the atrophy fibrosis phenotype, which is the loss of parenchyma. That's a permanent loss of parenchyma with parenchymal remodeling. And we're trying to develop a model that explains this. We started out in mouse models where we had isografts and allografts. And when we looked at them, we found that there was an injury response in we called these injury and repair-induced transcripts that peaks around day 3 and later ones that peak around day 5. The characteristic of those is they come up, they go down very slowly, and they may never completely return to normal. And they're reinduced when rejection starts in the allograft. So the isograft follows this curve, peaking around day 5, like wound repair, and then slowly resolving. And so that's the isograft. The allograft takes off as T-cell mediated rejection occurs and induces more AKI. There's a set of transcripts which actually only reflect the acute phase response. They're induced in the transplant kidney. They're also induced in the host kidney if you left the host kidney in. They peak around 24 hours and then decline. And as rejection occurs, they never come back with rejection, so that they are quite different. So the kidney that's transplanted, the heart, it has two kinds of molecular information. It has acute phase response information and it has injury information. So we wanted to make sure that we had the injury information. So then we have taken that into the human and established the injury and repair-induced transcripts in the human. These transcripts are we trained by looking at pristine biopsies, protocol biopsies of kidneys in the first 6 weeks versus indication biopsies of kidneys with GFR, abnormalities, but did not have rejection. And what came out was a set of transcripts. When you injure a tissue, you get tens of thousands of transcripts changing. There's a gigantic hierarchy. We talk about the peak of these, but never forget that there's also a very wide range of changes in that tissue. And the peak of those we've used as a useful concept called the IRATs, or the AKI-induced transcripts, and then we just follow those. This is actually the strength of association. There's up and there's down, and down is equal to up. There are as many transcripts lost, and this is why the function is down. Okay, the transcripts in the acute response, the acute injury response, wound repair in the organ, and some of them are typical, they have some interesting properties. They're increased in isografts and in ischemic injury models, so that they're the typical transcripts that you get in ATN models by cross-clamping. They're increased in human kidney transplants with AKI, and they correlate with the loss of GFR and the subsequent recovery of GFR. They're not predictable by histology lesions. There's no lesion which tells you this. This is histologically silent change in the tissue. It can be seen by confocal microscopy, but not by routine histology. Many are also increased in the hearts. The wound repair response is shared across the organs to some extent. It's also shared in transplants with progressing diseases, so new onset of injury induces AKI-like transcripts. It predicts the loss. It's the best thing usually that we have, and we've done that with a number of different ways as the risk score. And so the risk score is best predicted by the acute injury genes being induced by a disease. And pathway analysis reveals cancer. Now, why cancer? Cancer is the wound that doesn't heal. Cancer is a wounded tissue. And so we share features with cancers. And it's also shared with developmental changes because in an attempt to repair the nephron, you recapitulate the development of the nephron. Interestingly, fibrillar collagen genes are not associated with fibrosis. Fibrosis is a protein state. The remodeling of the matrix is fibrillar collagen transcripts as you actively are remodeling, and that's associated with the acute phase, the acute response to wounding. So what we're doing now to try to incorporate this knowledge into the assessment of the individual at the level of the biopsy is we're using global disturbance, inflammation. Now that is a way of assessing the tissue. It's like looking at the microscope for the first time, seeing how abnormal the tissue is. And what we're using is gene sets associated with both inflammation and injury, because injury, wound repair, is always inflamed. It's always a mistake, too, to think that the inflammation causes the wound. The AKI-associated gene set, and we have classifiers for detecting the probability of fibrosis, which of course is absent in early kidneys and comes on later. But in addition, now we're applying archetype analysis, so we get the complete picture of that individual organ with injury archetypes. So let me show you that. We do the same kind of archetype analysis that we did for the rejection phenotypes, only now we're doing it, we get three archetypes which seem to explain the range of changes in an individual biopsy. There's healthy, there's atrophy fibrosis, and there's AKI. Healthy anti-correlates with any inflammatory lesion. It particularly anti-correlates with atrophy fibrosis. It positively correlates with function, and it's independent of time. You can be healthy at any time. Atrophy fibrosis is always a little bit inflamed. And that's one of the problems with making rejection diagnoses, because the changes are not completely specific for rejection, molecularly or histologically. It highly correlates with atrophy scarring. It weakly anti-correlates with GFR, and of course, it's induced over time. AKI is not associated with any lesions. It's not associated with atrophy fibrosis. It's highly associated with depression of GFR. That's the strongest association we have, and it tends to be a phenotype which is more dominant early than late. So this suggests a model of the injured tissue. In our case, the nephron. The nephron can be injured at any time and undergoes a wound repair response. That response is associated with the AKI transcripts and a little bit later with the collagen transcripts as it's remodeling its environment. Then a decision is made. That decision is, am I going to repair and come back online, or am I going to disappear into the atrophy fibrosis never land and eventually be completely remodeled out? That decision is probably made by issues such as somatic cell senescence, maybe some local issues like podocyte loss, perhaps the extent of injury. Probably it's governed to some extent by feedback within the tissue. So at that point, the tissue can either attempt full restoration, which is what happens in the mouse isograft, or it can head toward atrophy fibrosis. And when it goes toward atrophy fibrosis, after 4 or 5 months, a characteristic form of inflammation occurs, which has mast cells and plasma cells in it. And that can be very confusing because some people have thought that's adaptive immunity. Now, prognosis, and this is my last point. When we're trying to model prognosis, if you only include the rejection dimension, you get highly significant changes. But if you use rejection and injury, the injury is the dominant phenotype if you're projecting survival after an indication biopsy. So these are the archetypes. The best survival is with no rejection, and pretty good with T-cell mediated rejection. The mixed phenotype tends to be associated with some early loss. The worst survival is with the full house and late stage ABMR phenotypes. The early stage ABMR has a lag period of about 2 years and then follows the same curve as the full house. If you instead do random forests and you put the injury and rejection information at the same time, and the conventional information that we always know predicts survival at the time of a biopsy, you get proteinuria, GFR. That's very reassuring. If you didn't get those, you'd be worried. And now the best thing is the acute injury response, the IRATs. Interestingly, in that model, the immunologic phenotyping doesn't have much of a role. It's how much damage the immunologic process is doing that determines prognosis. So this is what we've covered, and I hope you can see how we're moving toward identifying the full individuality of the individual, but moving toward meaningful, actionable categories out of that, which are predictable from that individual. So thank you very much.
So thank you very much, Phil, for this excellent presentation. So there's a question from a prominent person, Stefan Schneeberger, and he asked about the non-compliance phenotype. And his question is, how does the behavior translate into a phenotype beyond allowing rejection to happen? That's difficult, I guess.
Okay, so it looks like we're also seeing this now in hearts, that phenotype that is a tail on the distribution down in the TCMR area. That's associated, we were able to characterize the medical records of a section of these patients, and Joanna Solaris did a lot of this, as being recorded as suspected or proven non-adherence in the medical record, independent of our study. And so that's our definition of non-adherence. We find many people who have a form of severe T-cell mediated rejection, usually with early antibody-mediated rejection features, have been recorded as non-adherent. And so what we're doing actually is the data suggests that this is a group. We're going back to try to explain the group, and we put the label on as non-adherent, but that's where the label comes from. It comes from going back to try to explain why that group is regarded in the data as being separate from the T-cell mediated rejection group. Humans have a tendency not to experiment, and they experiment toward the end of the first year, and for over 3 or 4 years, we see a lot of these cases presenting. And after 15 years, they don't, because if they are experimenters, they're not getting to 15 years. So we see this phenotype presenting all too often, and I just don't, the question really is, how do you change the behavior? And maybe it's just not natural to ask a human to take medications twice a day forever.
I have just another question. So we all know, using conventional morphology, there's inter-observer variability, and I guess a major issue is also sampling error. Could you comment on sampling error using the molecular microscope?
So what we've done, and we did a lot of this with the help of our friends in Vienna, we have had ways of dividing the biopsy in two and finding that basically the molecular phenotyping is highly reproducible in two parts of the biopsy. These are IRB-approved consented patients, so the IRBs did not allow us ever to take 2 cores for this. So we know that there's a lot of biological reproducibility. The molecular changes appear to be a field defect, so even small pieces of tissue have all the information. Even though the disease is histologically appearing focal, it's not molecularly focal. And I think that that's why we have not seen much of a problem with sampling error until you get to the lung. But that's another story. It looks like the sampling error in the transbronchial biopsy is massive and has to be taken account into the math. So when we do encounter sampling error, and we can see it, like how much medulla and how much alveolar tissue there is in the transbronchial biopsy, we can deal with it by putting it in as another variable.
Maybe a last question. You did not touch the issue of endpoint analysis. I guess there's an intense discussion which endpoint should be used for clinical studies. Do you think that there could be an advantage of the MMDX, for example, in comparison to using eGFR slope in a study on ABMR?
So this goes back to my experience in all the registration clinical trials. Attempts to use eGFR in the individual patient have been dismally unsuccessful because of the variation. The variation in the measurement in the individual patient is greater than the potential variation in relationship to the effect of the drug. So actually trying to get individual endpoints in patients using those kinds of measurements is problematic. I would say if you're going to try for an effect on antibody-mediated rejection, use the molecular phenotype. Here it is it still there? Or is it down by 5% or 15%? We can tell you that. But trying to do it with function measurements when in the glomerulonephritis studies, in the glomerulonephritis registry in Toronto, they also tried functional measurements. They found that functional eGFR in the individual patient is all over the place. And that's not good for trying to get precision medicine.
Okay, so thank you very much.
Thank you very much. So Halloran, it's a pleasure for me to introduce our